students, and as far as I know, that this topic, gene technology, has been a really tough topic for all of you. Almost everyone hates it with their soul, and I can relate though. But let's get started, and we're gonna simplify each and every concept that this topic covers. First of all, we've got drug production from genetically modified organisms. Drug production from genetically modified organisms, it helps us, of course, to produce drugs or to help the body to survive at a better performance. So genetic engineering is a technique used to deliberately modify a specific characteristic of an organism. So all we need is a characteristic that it's missing in this organism, and I'm gonna get it from another organism. Here we have one organism, and that's another, excuse the drawing, that's another organism. I want this to have a specific characteristic, so I'm gonna pass this gene from this organism to that organism so that it's gonna have a similar gene that helps it perform a specific function. This technique involves removing a gene. Of course, that's what we've talked about, that we are going to remove a gene that codes for a desired characteristic, okay, for one organism, and transferring the gene to another organism where the desired gene is then expressed. For example, we have some rice that its nutrients are very limited, and it's going to cause malnutrition for people living in the area where this rice grows. So I'm going to get some bacteria, that this bacteria has a gene coding for vitamin A, and I'm going to pass this gene coding for vitamin A into this suitable rice, so that this rice is going to prevent malnutrition from existence. These people living in that area are going to survive better because the nutrients in the food they eat is way better than before. And that's all because transferring the gene into another organisms where we need this desired gene to be expressed. The genetically modified organism is said to contain recombinant DNA. And that's what we call the DNA that is almost a mixture. We can't exactly call it a mixture, but a recombinant DNA is like, I hope you guys imagine with me now that this is a circle, but I'm gonna remove part of it, and that's from another organism. This pink part, and this whole thing we can call the recombinant DNA. Of course, it isn't resembled in that structure, but it somehow shows you guys how a recombinant DNA is like. It's not only the blue part from an organism, it also contains part of it from another organism that is going to enable this thing to express a gene that we want. Okay, that's the recombinant DNA. And it's going to help in producing the genetically modified organism. Microorganisms such as yeast and bacteria, plants, animals have been genetically engineered to produce proteins used in medicine. And of course, that's been very perfect for such many severe problems, and it's going to be a huge cure to many things. Now let's move further into the genetically modified microorganisms, where we use restriction enzymes. I remember that you guys have heard of the restriction enzymes before, and these are the enzymes that remove the gene coding for a desired protein from an organism's genome and leaving the same sticky ends so that it's going to be complementary. Okay, so these are the restriction enzymes, and they remove the gene coding for a desired protein from an organism's genome. The genome is its DNA. The protein coded for here will be responsible for the characteristics desired in the genetically modified organism. For example, the ability to produce insulin. We have some kind of rat, and this rat is missing the ability to produce insulin. So we can let it produce insulin if we introduced a gene that we got it by the restriction enzymes, of course, that's going to remove the gene coding for the desired protein. I hope you guys got this concept, but we still haven't gotten started yet. We've got lots to do. Many copies of the gene are made using the polymerase chain reaction. Of course, I'm not gonna take a small, tiny gene from the genome of a specific genetically modified organism. This process itself is very hard and complicated. So once I get a small thing, I'm not gonna just use it in one process and then forget about it. I'm going to produce many copies of the same thing using the polymerase chain reaction. And as you guys remember in Unit 4, we've got also the polymerase chain reaction where we can produce more copies of the gene. I hope this was simple, and let's continue. The enzyme DNA polymerase is used to join free nucleotides into new strands of DNA that are complementary to the original strand. Okay, now we have one strand of DNA. I hope you can imagine with me. Oh, in here, we can say that this is some sort of a DNA strand. Okay, so we have here one thing and the other thing, and to join between these, it's gonna need some, of course I can't draw it, I'm sorry for you guys, but it's gonna need some sort of free nucleotides to join them. Okay, so forget this painting or like drawing, whatever. It's going to need free nucleotides. For further explanation, you can check Unit 1 for protein synthesis. We're gonna need protein synthesis and of course DNA replication. Free nucleotides into new strands of DNA in order to form the whole DNA molecule. Okay? These copies are inserted into small loops of DNA called plasmids. Of course, if we have some sort of bacteria, and this bacteria has plasmids in it. These are the small plasmids that the small loops of DNA, of course, the copies that we made is gonna be added to the plasmids. That these are the copies, and I'm gonna add them to the plasmid. And the plasmids are said to be DNA vectors. A vector can be referred to something that joins one thing or is the linking thread between two things. The enzyme DNA ligase catalyzes the joining of the desired gene into the plasmid vector. Okay, that's one good point, that we have the enzyme DNA ligase that makes them join together. It helps in joining them. The genetically modified microorganisms are grown in large fermenters, and that's of course a good point. We have to make them reproduce in a large number, because it's not going to be a small thing and we're just gonna cut off doing this, of course not. Containing nutrients, enabling them to multiply and produce large quantities of the new protein. I'm not gonna just do this new protein for one cell. No, of course, I'm gonna make a huge, large quantity of this new protein to be made. The protein can be yet isolated and purified before being packaged and distributed. I'm going to isolate it and purify it. Okay, these are 2 points. I'm going to isolate and purify. Isolate and purify. Human insulin and human blood clotting factors are examples of medicinal proteins produced by genetically modified bacteria. Can you imagine how bacteria that we thought are very tiny and small, and they could be meaningless for life, can help save lives by using this human insulin, the human blood clotting factors? That's insanely useful. Here is a diagram that's gonna help you guys recognize what we've stated, but in a more imaginary form. Like, you can imagine these functions of every part here and see it well. Here's the first part. This is of course the plasmid, which we called a vector. Okay, This plasmid vector is cut with the same restriction enzyme. We use the same restriction enzyme, which is here the endonuclease, that's its name, and that restriction enzyme of course cuts it and leaves this part I highlighted in blue, and these are the sticky ends that are complementary to each other, and they can just interact just fine and join together. Once they join using the help of an enzyme called DNA ligase, it's going to form this shape, which is similar, if you remember, to that circle I drew. To that circle, and then we remove part of it in pink. That's the whole idea in here, and it's gonna be joined like that, or whatever. Just forget about this part. This is what we call a recombinant plasmid. The term is so important, the recombinant plasmid, and it's inserted into the bacterial cell in here. That's the bacterial cell, and I'm gonna allow this to enter, reproduce, put it in a fermenter, and inside this fermenter you can produce a large quantity of that protein. Then I can do two things to that protein: isolate and purify. So I want to make a small summary for what we've said. First part is using the restriction enzyme in order to cut part of the DNA from the genome of the bacterial cell, okay, leaving of course something that we called sticky ends. Then after taking this genome, we're going to use polymerase chain reactions where DNA polymerase, or what we've called Taq DNA polymerase, actually that's what we're going to use in here, that is going to produce multiple copies of the gene. Multiple copies then are produced. These copies will be joined by ligase enzyme to the plasmid. Okay, that's for the plasmid, forming something called recombinant DNA. And that's of course, or recombinant plasmid though. It can be called either of these. Then the following step is placing it in a fermenter to produce more multiple copies of that protein. Then we can do two things that we've said: isolate the protein and purify the protein. Then it can be used in medicine. Thank you guys for watching. I hope this video helps.